Hello everyone, my name is Iman Ben Salam and today I'll be presenting my journal club presentation on the association between insulin resistance, type 2 diabetes, and the gut microbial diversity using a microbiome-wide analysis of the human gut microbiome. The aim of this study was to determine if there was a clear association between microbiome diversity and the development of type 2 diabetes using specific criteria. Research found that patients with type 2 diabetes and a higher HOMA-IR, which is a measurement of insulin resistance, were usually associated with less gut microbial diversity. Diversity was measured through alpha and beta diversity. They also found that the presence of butyrate-producing bacteria was associated with less type 2 diabetes and a lower HOMA-IR. The research also determined beta diversity and found an inverse relationship with insulin resistance. Overall, this study provided great information that the development of type 2 diabetes is strongly associated to an individual's gut microbiome health. The role of the gut microbiome in metabolic disorder developments has been studied in the past. I have specifically found an extensive study conducted by Harvard Medical School within some recent years. Their sample was much more diverse as the collected data was from individuals across multiple countries. But, similar to my article of choice, there was a strong association between microbial diversity and severity of type 2 diabetes. Studies like these suggest that the microbial-based therapies, such as the use of prebiotics and probiotics, may be successful in preventing severe diabetes. One of the main bacteria of focus when studying metabolic disorders is butyrate-producing bacteria. There has not been significant research on how these bacteria cause disorder to the insulin secretion pathway, but this molecule is important for anti-inflammatory properties that maintain gut homeostasis, and they're just important for overall gut health. I'll talk more about this later. Diabetes in itself is a chronic metabolic disorder that is characterized by high blood sugar levels. Individuals are diagnosed when they have continuous elevated A1C levels. An A1C level measures average blood sugar over 2 to 3 months, and it's a very commonly used blood test among physicians. There are many lifestyle factors that can increase someone's risk for developing diabetes. Some of these include insulin resistance, obesity, an inactive lifestyle, genetics and family history, diet, and even ethnicity. In fact, American Indians, African Americans, and Hispanics are more predisposed to diabetes simply because of their ethnicity. Some of these contributing factors are left in the hands of the individuals, such as activity levels and diet. However, there are some that cannot be controlled, like genetics. Once diagnosed, the physician will likely recommend weight loss, exercise, and a low-carb and low-sugar diet. Diabetes can be well controlled through lifestyle modifications, but most individuals will end up needing medications that control glucose or stimulate production of insulin in the pancreas. These kind of medications are successful in controlling diabetes because the overall disorder stems from the inability of cells to insulin, so they become resistant to insulin. This means that the glucose is not being absorbed or broken down well enough. I've presented a figure that will allow you all to visualize the mechanism because I know I needed some help to visualize it. Essentially, the body is not responding to the release of insulin, which can be caused by inflammation or other causes. And so once insulin signaling is impaired, glucose is not adequately absorbed by cells and used in metabolic pathways such as glycolysis. This leads to an excess amount of glucose being left in the bloodstream, which is how physicians are able to diagnose diabetes. This field of research is very necessary for the proper control of diabetes because diabetes can have many comorbidities and complications associated with it. I provided another infographic with some of the most common complications, some of them being diabetic individuals are more likely to develop diabetic retinopathy and glaucomas, which is why they have to get annual eye exams. They can develop diabetic neuropathy, which affects feelings of their extremities and can cause extreme pain. chronic kidney disorders, and most individuals with diabetes likely already have cardiovascular disease or will develop some cardiovascular disease in their lifetime. Diabetes was the 8th leading cause of death in 2021, and the number of diagnoses are rising every year, which means more research needs to be done for proper prevention and control of diabetes. Studying the gut microbiome can lead to developments in microbial therapy, personalized treatment, and new diagnostic markers for diabetes. It can also help us study the molecular mechanism of diabetes and how specific bacteria can affect it. In this study, the researchers sought to answer these two questions regarding the association of type 2 diabetes with microbial diversity and specific microbial taxa. So they specifically wanted to know which gut microbial taxa are associated with the development of type 2 diabetes, and is the development of type 2 diabetes associated with a more diverse or less diverse microbial community. This was a population-based study, so their sample consisted of 2 population-based cohorts in the Netherlands, the Rotterdam study and the Lifeline Steep study, each of which consisted of individuals over the ages of 18 and 45 who were at baseline living. This means some individuals could have been very healthy with no health complications, or could have regular health complications and may have taken medications from their day-to-day lives. Data from these groups have been ongoing since 2013 to 2014 for the Rotterdam study, and in 2013 for the Lifeline Steep study. Confounding factors including smoking status, dietary intake, physical activity, BMI, and use of medication were evaluated through questionnaires and interviews. It was very important for these facts to be taken into consideration because there are many confounders that can affect biological results that were obtained for this study. Microbiome data was collected via stool samples, which were sent through mail and stored at -20 degrees Celsius. They were obtained through commode specimen collection system and feces collection tubes for the collection of stool. DNA from these samples was isolating using diazorin and Qiagen DNA isolation kits. These kits work to purify DNA from biological samples through silica membrane-based spin column technology. Just like we've done in labs previously, it includes cell lysis and the washing and purification of DNA so that pure DNA can be used for PCR and other methods of sequencing and amplifying. The researchers then amplified the V3 and V4 hypervariable regions of the bacterial 16S rRNA and sequenced them using Illumina MiSeq. The process of 16S rRNA gene sequencing allows for researchers to examine complex microbial communities such as stool and differentiate a broad variety of taxa and species. Just like we learned in class, 16S rRNA is great for finding overall diversity and for sequencing large biological samples such as soil, water, and stool in this case. But some limitations of 16S rRNA sequencing is that we can't really determine the function of the taxa that we found. However, in this study, they weren't really worried about function. They were just worried about what taxa and how much diversity was present in each sample. So once identified, the alpha and beta diversities were calculated with multiple indexes such as Shannon index richness and inverse Simpson indexes. They also had a p-value cutoff of 0.5 and 0.005 to allow for significance. The beta diversity was used to compare species compositions from different samples. For example, comparing the samples from a healthy individual with no health complications and an individual with diabetes. Now we can take a closer look as to what associations were found. After analysis, it was found that higher microbial diversity is associated with lower HOMA-IR levels and lower prevalence of type 2 diabetes. HOMA-IR is a measure of insulin resistance, so a smaller value is associated with less insulin resistance. All models showed an inverse relationship, which supports the hypothesis of the importance of gut microbiome health for prevention of type 2 diabetes. There was 4 models total. In Model 1, an individual's age, sex, and the time their sample spent in the mail were taken into consideration. Over time, the researchers had decided to include most confounding variables, so Model 4 takes age, sex, time in mail, smoking status, education level, physical activity, alcohol intake, total energy intake, BMI, and the use of lipid-lowering or proton pump inhibitor medication into consideration, so this model was much more specific. In Table 2, we're looking at Model 4. We see a strong association with a p-value of 0.02. Although this is slightly weaker compared to the other models with less adjustments, the correlation was still very significant. The richness, which is the number of species in the gut, was also negatively associated with insulin resistance, suggesting that more microbial richness allows for lower HOMA-IR. There was the least amount of correlation between HOMA-IR and inverse Simpson index. This suggests that an even amount of microbial species is not very important for lowering insulin resistance and HOMA-IR. The association with prevalence of type 2 diabetes was measured by analyzing an individual's fasting blood glucose level or the use of blood glucose lowering drugs. The odds ratio was analyzed, which determines the association of the alpha diversity and the odds of developing type 2 diabetes. So in Table 3, which is the comparison of alpha diversity and type 2 diabetes, the strongest association was between higher species richness and a lesser likelihood of developing diabetes, as the p-value here remained consistent and low throughout all the models. Although the Shannon index was very strong in models 1 through 3, the significance was lost in Model 4 as the p-value becomes greater than 0.05. It was 0.06. There seems to be no significant relationship between prevalence of type 2 diabetes and species equalness, as the p-value is above 0.05 in all models under the inverse Simpson index. Overall, these two analyses showed a major trend of higher microbial diversity being associated with lower insulin resistance and less type 2 diabetes. Further analysis showed that specific taxa were closely associated with type 2 diabetes and HOMA-IR levels. A higher abundance of 7 taxa were associated with lower HOMA-IR levels, which can be seen in Table 4. Additionally, 5 other taxa were associated with less type 2 diabetes, and this can be seen in Table 5. All of the major bacteria that were found, all 12 of them, were butyrate-producing bacteria. In Table 4, we see very strong p-values of less than 0.01 and consistently negative beta coefficients, suggesting a strong association between taxa and insulin resistance. Table 5 also shows very strong significance between the 5 taxa and a lower risk of type 2 diabetes. So out of the 12 bacteria found, 2 of them have actually been studied previously. So Clostridiales and Clostridium have been previously associated with insulin resistance in other studies. This current study actually found 10 new associations with these taxa and insulin resistance that haven't been seen before. Since these haven't been seen before, this suggests the necessity for further research on the effects of bacteria and insulin sensitivity. However, there have been studies that do show a correlation between some of these novel associations. Cristian and obesity have actually been studied to have an association, and since obesity is a contributing factor to diabetes, this further supports our hypothesis on the effects of the gut microbiome and diabetes development. The importance of butyrate in gut health stems from its synthesis through fermentation of dietary fiber. So it's made in our bodies when we consume dietary fiber. So butyrate is a short-chain fatty acid that can provide protection against colorectal cancer, strengthen the epithelial barrier, and restrict entry of pathogenic microbes. Overall, the presence of butyrate-producing bacteria within the gut microbiome is associated with lower insulin resistance and less type 2 diabetes. However, further research seems to be necessary for understanding butyrate's role in metabolism and the insulin secretion pathway. This next figure shows the major taxa found in the sample before researchers started analyzing alpha diversity and correlations. So, as we can see, firmicutes and bacteroidetes were far more abundant than the other phyla found within the gut microbiome. The ratio between firmicutes and bacteroidetes is actually a very widely studied conception, and it has been linked to metabolic health. The ratio has actually been used as a marker for gut microbiome health, which is very important for diagnosing and understanding predispositions. These results support the need for analyses of an individual's gut microbiome as an additional form of preventative health, as many health complications have been associated with gut microbiome dysbiosis. Overall, these findings support the hypothesis that there is an association between microbiome diversity and the development of insulin resistance and type 2 diabetes. Specifically, the suggestion that less gut microbial diversity is associated with higher HOMA-IR and insulin resistance. Additionally, the presence of butyrate-producing bacteria is associated with less type 2 diabetes and lower HOMA-IR. This study contributes to broader implications such as the potential for new microbial therapeutic strategies. The significance of the association between these two variables should urge physicians, scientists, and pharmacists to develop and study more microbial-based treatment for type 2 diabetes and its prevention. For example, fetal microbiota transplantations and the use of probiotics can greatly improve someone's microbial health. However, more clinical trials need to be completed for these to be successful for humans. This study also implies the possibility of using biomarkers as diagnostic tools for diabetes and other metabolic disorders. For example, periodically measuring the quantity of butyrate-producing species within someone's microbial community can inform them of their predisposition of diabetes or other disorders, and this will likely create more urgency for lifestyle modifications. Although this study produced significant association between microbial community and type 2 diabetes, we still don't really understand the direct causality between them. Further research is necessary for understanding the mechanisms of butyrate and its role in insulin sensitivity or secretion. Furthermore, we should also focus on the effects of diabetes medication on the gut microbiome and make connections to other conditions. I have made several new conclusions and have had previous conceptions reinforced by the results of this study. I've always believed in the prioritization of lifestyle modifications in healthcare for the prevention and treatment of chronic diseases. As a pre-medical student, I find this to be something I'm very passionate about. However, I do also understand the benefits, success, and science behind medications and why they work for certain individuals. However, prioritizing microbial health through dietary changes and lifestyle modifications, such as becoming more active, seems to be a more efficient method for treatment, as it's something that's much more attainable than medication for some individuals. This study and other similar ones are important for improving personalized treatment of diabetes, because nowadays we're only using A1C and glucose levels to diagnose individuals. There's only a certain amount of treatments available, and some people may not tolerate all of them. Overall, this study was very well and had a very good population size and results, but I do believe there were some limitations. This study was a population-based study, so it had a great sample size and was very generalizable, but only to one demographic, country, and environment, which was the Netherlands. I think it's very important to also study microbial trends of other demographics, such as those I've mentioned previously that have a greater risk for type 2 diabetes based off of their ethnicity. In conclusion, I do believe understanding the role of gut microbiome within metabolic disorders is crucial for advancements in treatment and prevention of chronic diseases. These are some questions that I believe will guide discussion within our group, and I hope you all enjoyed my presentation. And here are my references. Thank you very much.